since then. And over to you, Frikkie. Yeah.
Thank you. So I've been introduced. This is not the title of my PhD project, so I'm going to talk a bit about the group that I'm working on and then do a case study on my project. But really it's been about the reason you mentioned, my DNA was ready to go. I spent 3 months optimizing a high molecular weight DNA extraction protocol for these little guys. And it's a genus that's been vastly understudied from a genomics perspective. The taxonomy has been well done, and it's a fascinating genus. So let me start to introduce them to you. When people hear Oxalis, they've got a very high reputation of being cosmopolitan weeds. We've got Oxalis corniculata. I remember the first time I went to Anya's lab, just walked out the door and there's a pot plant inside Diplomix's building, and I'm like, that's Oxalis corniculata. It's a weed all over the world. And that's often the identification that's going along with this genus. It's a reputation that's been built up. They're known, there are at least 5 very well-known weeds across the entire world. But there are notable exceptions in this genus to this weedy nature. And I'll introduce you to the other 500 species that are not known invasive global weeds. And we've got a very unique situation here in the Cape, one of the big diversification centers for this genus. Described, of these 500 global species, about 230 described, endemic to the Cape mostly, extremely morphologically variable group. And they're such a wonder to work on. I think the biggest thing about this, one of the biggest interests for this group, is the exceptional morphological variation that they've displayed. So for evolutionary biologists, it's a really fascinating system to ask evolutionary questions, the mechanism driving not just diversification of this group, but why we have these really stark morphological variations. I mean, you would not think these two do even belong in the same genus. And it's been a recurrent story that both within and between species we see morphological variation, but also variation in terms of the soil types, specialization of different species. We've got some that are moss seep specialists and some that are generalists across sand, fynbos, aquatic species like that. But what really fascinates me about this group is a unique combination of evolutionary traits that aren't necessarily unique to any plant group, but the combination of them are definitely unique in Oxalis. Starting off with nitrogen fixation, there are a host of range of different plant families and plant groups that form symbiosis with nitrogen-fixing bacteria or other groups. But Oxalis is one of these that has endophytic symbionts that are carried in those little channels that you can see on this seedling transect, and they're inherited through the seeds from parent to seedlings. And that's one of the traits. And it's not unique to Oxalis. There's a whole bunch of species that does this. But then we get to a different trait, Oxalis. It's an extremely polyploid-rich group, which, once again, it's not a very funny thing. There are many plants and animals out there that produce a whole bunch of polyploids. But in the context of Cape speciation and diversification, it's actually quite unique. So majority of the groups in the Greater Cape floristic region, which we all know is one of the largest diverse floral kingdoms in the world, clades and lineages are often polyploid poor. So they've got 1 or 2 cytotypes. And then Oxalis comes along. And we've got both within and between species variation. So my system, for example, Oxalis obtuse, has up to 13 cytotypes. And this is recurrent between and within species. Seed recalcitrance, so these are, for those that are not familiar, this is a trait in plants. A seed is shot from its capsule, and that plant immediately starts grow. Within 24 hours, you've got a seedling developed. And seed recalcitrance, once again, not a unique trait in any plant group, but it is quite a rare trait. I think less than 15% of angiosperms have evolved seed recalcitrance. But then I want to focus on these two, which for me is absolutely fascinating. First one is the evolution of bulbs. And for those that are not familiar with plant taxonomy, there are two vast groups. We call them monocots, so grasses and crop plants, and eudicots, things like camel thorn trees and woody shrubs and trees. And they've diverged very long ago, a couple of 100 million years ago. And bulbs are a unique geophytic form of storing food, typically associated with monocots. Oxalis is a eudicot plant, and at least to my knowledge, one of the only groups that's formed a true bulb, at least in the morphological sense of it. And then tri-styly. So that's really one of my focal points, and I'll talk about that a bit later. But the evolution of tri-styly, one could almost equate to the evolution of different sexes in humans, so having male and females. Plants can also develop sexual systems or these polymorphic systems. And tri-stylies essentially evolved for plants to have preferential mates with other morphs to avoid selfing and inbreeding depression. So this really makes Oxalis as a system an ideal model to test a whole bunch of evolutionary questions that one can relate to not just Cape evolution, but overall organism evolution. Things like, how do polyploid genomes occur? Why are they still around? Do they contribute to speciation? I'll get to conservation in a bit, but things like these weird morphological traits. Why is bulb selected for typically in monocots, but suddenly we've got a eudicot? Looking at questions about seed and germination strategies, speciation in the Cape, plant associations, both with bacteria and different fungus, to the sense that there's even a sexually transmitted fungus in some of these tri-styly systems. Plant mating systems, so evolution of these complex polymorphisms, have been a question of high interest for most evolutionary biologists out there. And then conservation. Conservation is one of the big drivers. Like I mentioned, there's 230 described species in the Western and the Northern Cape region, most of them endemic. And about 70% of them are data deficient. And of the ones that have been assessed for the red list, most of our aquatic or endemic species are endangered or under threat of climate change for future endangered. And that brings me to the work that I have been doing. So before one could actually think about creating a model system, you obviously need a reference genome. And the point is, the reference genome is a starting point to really interrogate these evolutionary questions from a detailed perspective, doing comparative genomics. And there's been no genomics work done on this. The work that has been done on Oxalis has vastly been phylogenetic, and there's been some plastid genome assemblies, and there's been some transcriptomic work. And in all of the decades and centuries of Oxalis research, that's what we have. So I set out to first build the first reference genome for this group. It was quite a challenge. I spent about 6 months optimizing a high molecular weight CTAB DNA extraction method, and then spent another 5-6 months figuring out how to phase these assemblies for a highly heterozygous individual. But in the end, we ended up with a very good assembly of an N50, a scaffolded size of 2.7 megabases, about 270 contigs, and a BUSCO completeness score of 97%. So this is an amazing starting point, right? We've got a reference genome. But what does a reference genome really mean unless you've got something to compare it to or something to build further from that? And that is where the genomes comes in that we've been doing with 1KSA, that they have sponsored. So initially when we got this question, we were like, well, we are a very small lab, we're limited by funding, and suddenly we're giving the opportunity to have 10 genomes sponsored for this system, so we need to make the most of this opportunity. We really went and looked at our taxonomy, so we chose genomes that are taxonomically representative of this group, of some kind of evolutionary interest, of conservation interest, of horticultural interest, and we also included the various floral morphs of that system, and I'll talk to why that is of interest to me a bit now. So let me introduce you to my plant. This is Oxalis obtusa. It is a widespread common species. It is one of those littered with polyploids, but we chose a diploid. We confirmed ploidy beforehand through flow cytometry for obvious reasons. But what really is amazing, it's also a morphologically variable species complex, and it's one of those issue trouble groups inside of the Oxalis phylogeny. But now I need to introduce you to the mating system of not just Oxalis obtusa, but of the entire genus. So the South African group and most of the other Oxalis groups out there have evolved a system called tristyly, and tristyly consists out of 3 distinct floral morphs in a single population. What's fascinating about this system, it's not perfect, but you can only get viable seeds by taking pollen from the anthers of the matching level from other 2 morphs to place on the stigma of the 3rd morph. And it's a way that small populations in plants can effectively increase outcrossing, increase genetic variation, and avoid selfing or mating with a closely related individual. Work on tristyly has been going on for about 70, 80 years. Most of it's involved in crossing experiments, theoretical data, and only recently have we had the molecular capability to actually assess the genetic architecture, but not just of tristyly, but of these plant mating systems that often bring out different floral morphs, of which tristyly tends to be the most complex. So let me quickly introduce the theoretical work behind it. Theoretically, the 3 morphs are determined by these 2 loci, the S locus and the M locus. And for each, there will be a dominant and a recessive haplotype. And in Oxalis at least, and other tristylis systems, it's also been confirmed somewhat, the S locus is epistatic over the M, which means that it's preferentially expressed. So you can have a whole bunch of combinations, and eventually that's how you arrive at the 3 different floral morphs. Here I need to take a quick pause because I've had a whole bunch of conversations with molecular biologists, especially in my methodology in trying to find these loci, because loci are notoriously difficult to categorize and find with high confidence. But because these loci are so determinant of the morph, there are very predictable patterns to look for. In a monocot group that also have tristyly, they've classified, this paper came out 2 years ago, it's published 1 year ago. They've classified these loci at a molecular level and found that there are a couple of unique genes that's situated on the one allele of this locus that's absent on the other. So that's one of the general criteria of these loci, is that they are hemizygous, meaning that on an individual of one morph, the genes are present on the one haplotype and absent on the other. And that's one of the key patterns that we're looking for. The second thing that we can look for is, because these loci are predictable of morph, they are usually located on a chromosome that's suppressed for recombination. So no recombination happens at a molecular level. And as a result, you've got an accumulation of SNPs or mutations in those regions that eventually can be picked up at a genome-wide scale quite easily and quite predictably. But it's quite convenient to have a large lab with 4 different technical support labs, sequence 20 individuals of all 3 morphs, do chromosomal scale assemblies for each one of those individuals, because that's what this study has done. And then you sit with a smaller lab which do not have that capabilities. And I think that's really been one of the key successes that 1KSA has contributed to my project, because pulling the 10 genomes that I got from 1KSA with the other 2 genomes that we have sequenced, we end up with 12 genomes representative of all 3 of the floral morphs. And what we then can essentially try and do is group the genomes. Even though they are different species, they still have the same reproductive system. It's probably conserved, so it's inherited across the genus. and we can pool these species by their morph and do these FST or calculate the comparative FST value. And you can say, well, firstly, it seems like your SNPs are occurring across the entire genome. And secondly, you can't just be sure that a SNP that's associated with one morph is actually determinant of tristyly. And both of those are correct. But I can explain them. The first one is we still have a highly fragmented assembly. We've got about, for the reference plant, we have 280 contigs. For the reference that we use to map all the reads against, we have 480 contigs. So what's most likely going to happen when we do synteny analysis is hoping to find that all of those contigs associate and are expressed together. So they've got some kind of linkage going on there. And the second thing is we have very few SNPs. But it's sort of a bit of a yes-no argument because we grouped different unrelated species into these morph classes. It's very likely that SNPs that you find highly unique to the morphs might be associated to those regions. And the only way that we have actually managed to find these out has been by these 10 sponsored genomes from 1KSA. This highlights regions of interest across my genome that I can go and investigate. But really what this talk is about is trying to show that even at small systems with small labs, the contributions that 1KSA has made and that what we're trying to build is what is the genetic foundation that we've essentially created for this group. I've shown this one case study for my project that looks into this floral polymorphism. But already with the reference genome and these 10 other genomes that's got sequenced, we have 3 other projects that are already utilizing these genomes or potentially utilizing these genomes to look into all of these evolutionary questions, things like polyploid evolution, conservation orientation, and population genetics. And it really just highlights the importance of creating a foundation on which molecular studies can be based for these small systems, unknown systems. So what I have done, we have an assembled and annotated reference genome. We have assembled 3 floral morph genomes for the same species, so representative of this mating system. And we have floral transcriptomes for these genomes. And from 1KSA part, we also have 9 other Oxalis species that are taxonomically representative, all of them diploid. And I think that is a very solid molecular foundation, a starting point for this group to move research forward and answer a lot of these questions that we don't have the resolution to answer at this point. So I'm going to leave you with this little slide. Thank you for your time. Any questions?